Assalamu alaikum, dear fellows, स्टूडेंट्स एंड सब्सक्राइबर्स। इस वीडियो में आज हम डीएनए रेप्लिकेशन का कंप्लीट प्रोसेस डिस्कस करेंगे कि डीएनए रेप्लिकेशन सेमी कंजर्वेटिव कैसे है। तो आइए चलते हैं हम अपने लेक्चर्स की तरफ।
DNA replication. DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of four chemical bases, represented by the letter A, Z, G, and T. The two strands are complementary. This means that wherever there is a T in one strand, there will be an A in the other strands, and wherever there is a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. Let's determine how each strand of DNA is replicated. The first step is DNA replication is to separate these two strands. This unzipping is done by an enzyme called helix and results in the formation of a replication fork. The separated strand each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. RNA base. This enzyme makes a small piece of RNA called the primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in 5' end to 3' directions. The other strand, the lacking strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Agasagi fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a sort of row of DNA bases in the 5' and the 3' directions. The next prime is then added further down the lacking strand. Other Agasagi fragments then made and the process is repeated again. Once the DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Other DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as a semi-conservative because each DNA molecule is made up of one old conserved strand of DNA and one new one.
Dear viewers, आपको ये वीडियो अच्छी तरह समझ आ गई होगी। अगर इसी ही लेक्चर के रिलेटेड कोई क्वेश्चन है तो आप कमेंट्स में पूछ सकते हैं। चैनल को सब्सक्राइब करें। मिलते हैं नेक्स्ट वीडियो में, तब तक के लिए अल्लाह हाफिज।